So we want to sequence a lot of genes. We want to sequence 2,000 genes. So we want to repeat that experiment 2,000 times. Now the problem is that just by chance, you're going to see differences between the people that have respiratory distress and the people who don't have respiratory distress. Okay? So what you want to do is you want to sequence enough individuals, and then you can set your cutoff of the number of mutations, the difference, very high, so that you can say, look, this would never happen by chance. So if you're going to set your cutoff high, you've got to sequence a lot of individuals. So this tells us the power of our detection. So this curve, all you need to really know is that we would detect almost all of the mutations that we expect to observe if we sequence roughly 300 individuals with respiratory distress syndrome and 300 individuals without respiratory distress syndrome. There's a little caveat there, which is we're assuming that each of these mutations has a relatively large effect. Okay? Okay. The problem is, if we were to do this by standard methods, again, all we're doing is sequencing, but if we were to do this by standard methods, it would cost $18 million. Okay? So that's a bit out of our lab's budget, but we're really fortunate actually because this is an extremely exciting time in the field of genetics. This is an extremely exciting time in the field of genetics. And the reason why is that the cost of DNA sequencing is dropping exponentially with new technologies. Okay? So how many people are familiar with Moore's law? Anyone familiar with Moore's law here? Okay, so what is Moore's law? Moore's law states that the number of transistors, it's about computers. Okay? It's about the field of computers. And it's the reason why we've had the computer revolution. It's the reason why we've had the internet revolution. Okay? And it states that the number of transistors on a microprocessor doubles every 18 months. It was formulated by Gordon Moore of Intel. Okay? And so that means that it's an exponential growth. Okay? And what it means is that your computer that you bought 2 years ago is now basically a piece of junk. But what that's done is that incredible growth in the computer has made computers more and more useful, and we've been able to attack ever more challenging problems with computers. Well, DNA sequencing has been undergoing a similar exponential growth. So this curve here is Moore's law. This is the number of instructions per second per US dollar as a function of time. Okay, and this is a log scale, so that means exponential growth. All right. Here's the cost of DNA sequencing. All right. So it's actually been tracking Moore's law for quite a while, but now you see this big change in the slope, and we're passing up Moore's law here. That is next generation sequencing technology. Okay. And so that is what's making this $18 million, what did cost $18 million, now costs, what is it, $120,000? A sum that fits in a CDI budget. Okay. Okay, so how is this achieved? If you want to go into the details of how this is achieved, I'm happy to talk about it offline. But really, the principles were actually the same principles. So this was achieved by a large number of researchers working, a large number of different researchers actually simultaneously, and there's now commercial technology that makes this available. But this particular slide is from Illumina, which is a company that is working on next generation sequencing technology. But the concepts of next generation sequencing technology are actually exactly the same as that of the microprocessor, which is integration, miniaturization, and parallelization. Okay? So what that means is that all of the steps of the sequencing reaction are done in an integrated fashion, and they're done in a very small volume, and they're done in parallel. So see these spots here? Each of these spots is 1 micron in size. Okay? And each of these spots is a different sequencing reaction. So you can fit 100 million of these spots on a microscope slide. Okay? So you can do 100 million sequencing reactions in parallel. All right? Whereas with the old technology, you were doing 384 in parallel. Now you're doing 100 million in parallel. Okay, so this is what's catalyzed this. Okay. Okay. So this technology is very interesting, and one problem though with the technology is it's still too expensive to sequence 600 genomes. So we still can't sequence all of the baby's genomes. What we want to do is we want to grab out these 2,000 genes and then sequence those things. So 2,000 genes is actually 20,000 different parts of the code because each gene is 10 different non-contiguous sections of the code. So if you wanted to, you could grab out all the different parts of all the different genomes, and that would be fine. But the problem is you've got to grab, that means you've got to grab out you've got to do a chemical reaction to grab out each little bit of the genome. And that means you've got to do 6 million reactions to do this, okay? And so Todd, who was working in my lab, didn't want to do 6 million reactions. And so he and some other people thought of ways to reduce this number. And there are different ways to reduce this number, but the strategy that he struck upon was he could pool all the individuals and then just do 20,000 reactions. And then if he could figure out a way to read out the sequence from this pooled sequencing, then he could still get the same information that we needed before, which was that list of mutations in the cases and the list of mutations in the controls. Okay? So the idea would be you pool all of your people that have RDS, all of your kids that have RDS, and all of your kids that don't have RDS. And then you sequence them individually, and you look for places that have mutations. All right? So this was a good idea, but the problem is that imagine you're going to sequence one base pair in the code. Okay? So you sequence it 299 times, and you get 299 times you get the reference base. All right? But on the 300th time, you see, oh, there is a mutation there. You see, this is actually what you're looking for. You found a mutation on the 300th time. The problem is that you don't know whether that's a sequencing error because the method's not perfect, or if that actually represents a mutation in your individual, which was at 1/300th of your sample. Okay? So this graph sort of illustrates the problem. So for example, if I put the fraction of reads that have a mutation on the y-axis, and on the x-axis I have the position in the code, so this would be position 100 in the code, I see these spikes. So for example, at this position, I see about a little bit less than 1% of my sample has a mutation at that base, when I sequence that same base again and again and again. Okay? Now, the problem is that you can see here the problem with the signal and the noise, the sequencing noise. Wherever I see these arrows, that's where there actually is a bona fide mutation in this sample. And all these other peaks are noise. Okay? So Francesco Valagna, a graduate student in my lab, came up with a solution. And I'm not going to go into the details, but he used some tools, mathematical tools from information theory, to essentially pick out these peaks from the rest of these peaks. Okay? And so, so the question is, does it work? And it works pretty well. It works pretty well. We sequenced the surfactant protein C gene, okay, which was previously sequenced by Sesh and Aaron by the old method, okay, and we compared it to this pooled method, okay, and we found 7 of 7 of the known SNPs. We found an additional 14 predicted SNPs which were in regions that weren't sequenced. We got 2 of them were wrong, but we can go back and check these once we find these. So this was actually a pretty good performance. And the cost of this pilot for sequencing this gene was about 1/100th of the cost of the original project. Okay. So our conclusions: the strategy is sound. We think we can do the project exactly as proposed. We think we can do it on the scale that was proposed, and we think we can get the statistical significance that was claimed. Okay? And we hope that we're going to find a number of genes that predispose to respiratory distress syndrome. Now, what happens if we don't? Okay? We can do checks to make sure that all of our technology was sound. But if we don't, actually, I think that will be very interesting too, because we've really been classifying, we've really been ruling out what's going on in terms of why we think people get disease. And if we're not able to find it by this method, okay, then something very interesting genetically is going on. And so, of course, one study isn't going to do it, but the first data point that even if we can't find disease genes, the first data point that says, hey, you're looking in the wrong place, will also be very, very informative. Okay. So we're going to go and, so our next step is to actually go and start sequencing the 2,000 genes. Our time frame is 18 months. And I'd like to thank, these are the people who did all the work. I mentioned our collaborators, Todd Drewley, Sessions Cole, and Aaron Hambus, and Francesco, who came up with the algorithm. And these are the people in my lab who help with the sequencing. Thank you.